here so everyone can hear me in mind.
All right, I'm going to go ahead and get started. It's my pleasure today to introduce Athena Nghiem. Athena comes to us from Wisconsin, where she's been for the last year, starting up a research group in geosciences. She is someone who she graduated about 4 years ago, as I was tallying in the math in my head, um, at the end of the, you know, normal times before the pandemic, basically right as the pandemic was whirling up. And her thesis here was really related to arsenic in Southeast Asia and a lot of data science approaches to try to understand the connections, between sites and scaling environmental measurements that we make in geochemistry to larger environments. After that, she went to ETH Zurich, where she worked with Lenny Winkel on related work and some other stuff, especially related to selenium. And now she is back here in the States again at Wisconsin, where she's um, just doing a whole bunch of fun things that I'm just learning about today. And with that, she's going to share some of the work she's doing on arsenic and that she's been working on over the last few years. And I'll let her kick it off here. All right.
Okay, my mic is on. Thank you everyone for coming here and either braving the rain in person or online. It's wonderful to be back at Lamont. I haven't been since I defended my thesis, so this is definitely less stressful and really exciting to show what I've been up to in the last few years. So as Ben mentioned, I really got my start in arsenic biogeochemistry here at Lamont and also as part of the Columbia Superfund Research Program as a trainee. So it's really wonderful to be back here and show some updates on my research. And my research today will look at biogeochemical cycling, but also different reservoirs for the redistribution of this threat to human health. So let's see. I think this disconnected, but it's okay. I'll just go off my laptop. So here's our fundamental water cycle. And just like water in the environment that goes through different reservoirs, arsenic also has different reservoirs and different cycles which it goes through in the environment, ranging from terrestrial to the ocean to the atmosphere. And one of the primary reasons why I and a lot of other people study arsenic is because it's a trace element that's naturally present in the environment that undergoes this biogeochemical cycling, but it also carries a really significant threat to human health because of its frequency, toxicity, and thus potential for human exposure. And I'm sure the public health scientists can tell you a lot more about that side. Different pathways, though, to briefly recap for exposure to arsenic include the consumption of naturally occurring or geogenic arsenic contaminated groundwater, as well as when this groundwater is used for irrigation, it can be uptaken in the soil environment as well as plants, and then lead to the consumption of arsenic-containing food. And finally, also the inhalation of atmospheric arsenic, which many might not think about, but is also present in the environment. So here we have different sources and sinks of arsenic in the environment. In particular, we know that groundwater is a naturally occurring source of contamination, but it's actually our use of groundwater modified by our anthropogenic flux that actually causes its exposure. Thus, understanding arsenic distribution in the environment is the very first step to understanding exposure to arsenic in the environment. So today in my talk, I'll be discussing some investigations of arsenic distribution. First, I'll talk about our topic that we know and love really well, which is groundwater arsenic contamination and groundwater pumping on a localized scale. But then we'll zoom out a really big into the atmospheric side and take a peek into the arsenic distribution in the atmosphere. So those are our two topics for today. Sure. The numbers, great question, live audience question. These numbers should be either the flux or the reservoir amount. And oh, yes. So live audience, here is, it's gigagrams of arsenic per year for the flux. And this was a review paper, and I will tell you why these fluxes are not exactly accurate later on in my. Yeah, that's a great question, Steve. Yeah. Yeah, I will get to the biosphere later in the arsenic part. I'd love to talk to you more about that too. Yeah, yeah, arsenic, betaine as well. Yeah, okay. Okay, so now we're going to go back to arsenic and groundwater before we reach the biosphere. Just a brief recap for those of you who don't think about arsenic all the time. Arsenic released to groundwater a lot of times is commonly attributed to the microbial reductive dissolution of arsenic-bearing iron minerals. Here you have your iron minerals with arsenic sorbed onto the surface. They undergo reduction coupled to the oxidation of organic carbon, and then arsenic is released into groundwater. Of course, if you remember from my PhD defense, there's a lot of factors that lead to this variability in arsenic, including hydrology, mineralogy, biogeochemistry. Of course, you can make it even more complicated. That really creates all of this variability at multiple scales. So while I was in my PhD, I had this great opportunity to be part of the USGS PAL Center Working Group with Ben and also Mason Stahl, who is at Union College, and there was a compilation of arsenic concentrations in groundwater across various studies and field surveys. And anything that's not in this blue color is above the World Health Organization and EPA maximum contaminant level of 10 micrograms per liter. So when you're zooming out at a large scale, it's hard to see the inherent variability of these arsenic concentrations. But when you really zoom in, you can see how heterogeneous this is. So your well will could be low in arsenic, and then your neighbor's well might be very high in arsenic. And so this is some of the variability that's characteristic of groundwater arsenic concentrations. In addition, where you do have low arsenic groundwater that is available, this may be jeopardized by increased groundwater pumping. So the infiltration of arsenic into low arsenic aquifers could also occur. And as we know from a lot of previous work that was done in Vietnam, including this study by Lex a while back ago now, we can have high arsenic water infiltrating into low arsenic aquifers. This is also a scenario that is also present in Bangladesh, and it kind of begs the question, what mechanisms and biogeochemical processes occurs when this happens with your high arsenic going to low arsenic aquifers? So this was actually some work that came out of Lamont that was done by Rajiv, another previous grad student of Ben and Lex, who looked at this exact scenario in a column experiment. And then I had the opportunity to model it at the very end of my PhD, and it finally came out after I defended my PhD, which is really exciting. But what we found in this paper was there was a new mechanism that hadn't really been much considered. So instead of this typically proposed mechanism that I just simply showed you earlier, we have an alternative mechanism in which instead of on the left-hand side was that iron-reducing bacteria story, we also thought about the influence of sulfate-reducing bacteria and the sulfur cycle, and in particular, how it could release these other species that I show you right here, which are also dissolved arsenic species, but a different speciation that could lead to faster mobility. So essentially, in this column experiment, we found that these species were important to explain these mechanisms. And this is because these species, which are called thioarsenic species, have low or no sorption to iron minerals compared to arsenic oxyanions. So on the left-hand side, we have our typically considered model, and on the right-hand side was our new finding from this work. And it leads to faster mobility because these thiolated species no longer sorb onto our iron minerals, and this thus can lead to faster contamination. So in this scenario, we saw that there was a redistribution of arsenic, and that limited sulfate reduction is sufficient for rapid contamination of groundwater with arsenic, even in aquifers with these oxidized iron minerals, as in this scenario. And thus, this could lead to faster redistribution of contamination and increased public health risks, especially as you increase groundwater pumping. So that was the conclusion of my time and work at Lamont. And to follow up on that, we still had a lot of open questions on this scenario and these mechanisms. For example, we knew that this happened in our column experiment, and elsewhere in the world, one of the best scientists at measuring these species had found them, for example, in Bangladesh groundwaters. So we had some follow-up questions that we still were thinking about, including what about similar scenarios elsewhere? Can we actually measure these dilated species in similar environments where we have a high arsenic flowing into low arsenic aquifers, for example? Can we apply a similar biogeochemical network to different field sites? And can we use total concentrations to our advantage? Because I said we were measuring these species were important, but measuring species is different from measuring total concentrations. So the first question is, could we measure dilated species in similar environments? And this was done with Britta Planner-Friedrich shortly before she passed away earlier this year. So it's very meaningful to be able to present this work. So let's go back to Vietnam here. Here we have some groundwater arsenic concentrations that came out of the study of Lenny Winkle, as Ben mentioned, who is at ETH Zurich, and I did my postdoc work with her. In this previous study, she found that Pleistocene aquifers were increasingly contaminated, and this was thought to be driven by kind of this groundwater pumping that had occurred in this region. So kind of a similar scenario to the one that we were setting up in this column experiment. So with that, we wanted to do a untargeted sampling campaign to measure these dilated arsenic species to see if the scenario we presented in the column experiment was relevant in real life in this mega kind of real life example. So coincidentally at the time, Lenny had a PhD student who was doing some groundwater sampling anyway. So I was like, this is a perfect opportunity to do some speciation analysis in collaboration with Britta. So you see that the points here were not because I selected them for any particular reason, but just because those were the samples that were remaining to be taken in our study. But you can see that this kind of follows the distribution of samples that are in Hanoi where this contamination had been previously found. So here are the preliminary results. On the left-hand side, you see a violin plot, which is like a box plot with a histogram of total arsenic concentrations, which go up really high to up to 400. And on the right-hand side, you see the arsenic species. So there's different arsenic species, and this was measured with ICPMS. If we zoom in, we see that we have our thiolated species shaded here. And then we also have each of their names. So monothiolated, dithiolated, trithiolated, and monomethyl and thiolated species. And you can see that it's actually a really small percent of our total arsenic. So it actually doesn't make up that high of a percent of the total arsenic. Way less than we expected. Actually, there was more methylated species that were found in this region. So even up to close to over 8%, maybe up to 8% of our monomethyl arsenic species. So overall, we found that methylated species were found more frequently than what we thought of the thiolated species. Interestingly, methylated species also have lower sorption and potentially higher mobility onto iron minerals as well. So the speciation being methylated could also impact its sorption onto iron minerals. But we still had some remaining questions, mostly because from all these sampled locations that we took, these were the only ones that had our thiolated species. And another question that we had was about the actual method, because if I know a lot of you have done international field work, and especially with my time with Ben in Vietnam, we know that field sampling is super hard, especially when you also have to account for preservation of species. And specifically for arsenic preservation, for speciation, you have to do a lot of different things, including add iron complexing agent and flash freezing. You have to immediately flash freeze it in the field. Here we have a box of dry ice, and it cannot thaw during transport or speciation is not preserved. So we were thinking, is it actually the method that we're doing in the field that is not preserving these species or transport along the way, for example? That could be a reason why we were not finding as much thiolated species as we would have expected. So as I mentioned, it's super arduous. So from these preliminary results, another thing we could think about was the transformation of species due to our preservation protocol. So to test kind of these hypotheses about our preservation or whether the species was transformed, we then did another sampling campaign in 2024. I called this a targeted species a sampling campaign because this time we actually went to sites that we wanted to measure to see if the thiolated species could be found. So as I mentioned, what we wanted to do was check the preservation method and actually this time predict where thiolated species could occur. So with the data set that we had from 2023, instead of just randomly going to different sites and seeing if we could find the species, could we use the data that we already had to predict these species in certain locations where the species had been measured? And why is because, well, one way that you could do the predictions is thermodynamics, but thermodynamics were not found to match some experiments. And you could also, this could also be related to kinetic factors. So instead of actually using thermodynamics or kinetics to do these predictions, we instead did machine learning to predict arsenic speciation. So using the parameters that we had from the 2023 sampling campaign, which included total concentrations, ion measurements, dissolved organic carbon measurements, extra lat long measurements, we wanted to predict the existence of these thiolated arsenic species. And because we couldn't reliably trust the concentrations due to species transformation, we could just trust if it was measured or not. We instead just predicted a classification, whether it was measured, true, or false. So the presence or absence of these species. So what we did in our preliminary results, we found some useful variables to use for our prediction. Here, of course, we know sulfur is an important part of the sulfur cycling story. So the difference between true or false in our case is shown on the left. And then actually well depth was another interesting parameter that had some differences between whether the thiolated species were measured or not. So when we put this together into a random forest model, here's what it looks like. We had a training and a test set, and we also did some cross-validation. And you can see in this most important predictor plot that sulfur indeed is one of the most important predictors for making these species with that attached sulfur group. We can also see that methane, chloride, and phosphate are also really important. But to use this data, actually, we wanted to take the results after we trusted our model and then actually use them and go back and measure to see if our model was actually valid. So in this targeted campaign in 2024, we went back. Here's my wish list locations for arsenic speciation. As you know, you have a lot of samples that you want to take during field work. So what is actually possible is actually different from what you can actually get. I had some locations where I wanted to remeasure to check the method. I had the strongest predicted places. So those were groundwater where I was more confident in the prediction of speciation, and then some weaker predictions. Actually, we were only able to get the remeasured ones and the strongest prediction. Also, as you know, people who do field work in actually very urbanized areas, a lot of our groundwater wells were also disappeared after 1 year of field work. So that was also a very common thing to occur. But actually from these wells, we did confirm the method that 12 out of 12 wells, which did have thiolated arsenic species, were remeasured again the next year. And then in our prediction, 7 out of 7 were predicted to have thiolated arsenic and then were measured to have thiolated arsenic, which gave us confidence in a model and now gives us the ability to interpret the random forest results. Yeah, live audience question. Yeah, that's a good question. We only did a validation set for the ones that were predicted, so we didn't measure the ones that weren't predicted to be true, if that makes sense. But that's a good question. I should see if we measured any extra ones and then look at that data. So we only targeted the ones that were true in our modeling prediction. So here is the histogram. You can see that the percentage is still small though. So this leads to still some open questions. Is it just that the percentage is small because the well screens are so long in the area, and thus these kind of interactions or these biogeochemical mechanisms that are happening kind of are diluted with the overall groundwater that we're measuring? We're still thinking about that, as well as the importance of the methylated species, which we still see are present. So the main result of that work was that species besides arsenic oxyanions can lead to faster redistribution of contamination of groundwater. So just the emphasis on thinking about other species besides our typically considered ones of arsenic oxyanions. And that we should consider speciation in biogeochemical reaction networks, for example, when we do modeling work. And then a third thing, which we didn't have much time to think about, was to account for temporal variability in measurements of speciation. So in the work that I mentioned by Britta earlier, there could also be time series changes in total arsenic concentrations, of course, but also in the speciation of arsenic over time. And then lastly, we must consider the generalizability of biogeochemical reaction networks and how to more accurately characterize arsenic mobility and contamination. So that was the groundwater portion. Now we're going to kind of zoom out a bit more from thinking about local redistributions to our global redistribution. And this was also some of the work that I did at ETH, looking at investigations of arsenic distribution in the atmosphere. So I also know some of you think about the atmosphere as well here, but so you may already know that the atmosphere is a very globally relevant and dynamic reservoir, and arsenic can undergo long-range transport after emissions from multiple natural and anthropogenic sources. So there were some past estimates of the lifetime of arsenic, and it was estimated to be 4.5 days, and emitted arsenic can be redistributed from local sources to the global environment. So as compared to arsenic in groundwater, where your exposure is mostly from drinking the contaminated groundwater, arsenic can travel more far and from its source and be inhaled. So it's a very different type of exposure source. That's how long it would be, for example, if something's emitted until it's deposited elsewhere. So following the atmospheric circulation patterns. Yeah, it's similar to residence time. Yeah, it's just called lifetime in atmospheric science. Great question. So if you're thinking about what are the emission sources of arsenic, anthropogenic sources of arsenic to the atmosphere, of course, include coal combustion, smelting and metals production, especially copper and other elements, mining, burning for agricultural and waste incineration, burning of fuel and wood sources, previous use of arsenic as a pesticide for agriculture, and gas from landfills. Meanwhile, natural sources of arsenic into the atmosphere include, of course, volcanoes, dust and wind erosion, forest fires, sea sprays, and also biogenic sources such as biological volatilization from very long postulated terrestrial sources and more recently emphasized marine sources. And I'll get into this a little bit later. But to basically see the schematic, you can see the difference between natural versus anthropogenic sources here. So our natural in green and our anthropogenic in orange. So yes, coming back to the schematic on the front slides that I had, there have been a lot of different past estimates of fluxes for both anthropogenic and natural emissions. And this is one of the big questions that we had because they have such large ranges depending on how the estimate was done. So way back already in the 70s and 80s, there were a lot of box models that tried to estimate these fluxes from natural and anthropogenic sources. And the percent that you see on top of each of the box is the percent natural over the total emission. So you can see that there's large variability in both each of the estimates themselves and over time. And more recently, there has been more atmospheric modeling work to try to investigate these sources. But you can see, and I'll point out later, that they don't really have a lot of natural sources in their estimates, mostly only anthropogenic. So since these models have been done, there have been a lot of experimental and computational methods that have been greatly advanced, especially on the side of instruments, better measurements of trace elements, but of course we can also have more complex atmospheric model simulations. Especially in some of the previous box models that I showed you, some of the estimates were only based off of one value they found in the ocean, like one time extrapolated to the whole globe. So there's been a lot of progress in both aspects. If we first look to the better measurements of trace elements, we again go to thinking about speciation. We know, thanks to the great work in measurements, that there are better measurements of both total trace element concentration, but also much progress in speciation. So again, here are oxyanion species. And now I'll introduce to you some atmospherically relevant speciation. So in the atmosphere, we have our volatile or our gaseous arsenic species. We have arsine, monomethyl arsine, dimethyl arsine, and trimethyl arsine, depending on how many methyl groups they have. Trimethyl arsine is said to be the last stage of the Challenger mechanism. So biological organisms, for example, would emit this as the last, kind of the last species into the environment. And after emission or release into the atmosphere, they undergo oxidation and then are transformed into the oxidized versions of these species. And these species then are low volatility, so then they undergo deposition in the environment or sorb onto or condensate onto aerosols, for example. So in the previous group at ETH, there was a lot of work done on trace element speciation, influenced by long and short range atmospheric transport. This is the work of Dr. Esther Berninger, who is also in the same group at ETH, where she measured different arsenic species at this observatory in the Pyrenees in the French mountains, really beautiful, called Pic du Midi. She had a 5-year aerosol series, as well as sampling of rain and cloud water at high time resolution. So she measured the speciation of arsenic, but also selenium and sulfur. And you can see here that there is a large percentage of methylated arsenic, particularly the species trimethylated arsenic, which I mentioned was the last thing that biological organisms could emit. So this hints at a biological source for these arsenic species. Okay, now moving on to the modeling side. Before we had mostly box models or estimates of flux, but now there's, of course, more complex atmospheric simulations available. And the nice thing about kind of these atmospheric models is that they can simulate atmospheric chemistry, climate, and transport. And using proposed and known emission sources, these atmospheric models are then validated against kind of the measurements that I just showed you of aerosols and depositions. So this was a model that, for example, tried to do this with arsenic coming from anthropogenic sources and some volcanoes, but this model was transport only. So arsenic was only essentially modeled as PM 2.5 in this model, not accounting for any chemistry that occurred. And of course, models in general are only as good as the incorporation of the measurements and also the mechanisms that we considered. So now finally going back to the biogenic side, actually natural biogenic emissions of arsenic by organisms have been known for 100 years. If you know something about the history of the use of arsenic, there was a long time ago use of arsenic in kind of this green wallpaper that you see an example of, which led to the release of arsines, which is a really toxic gas, and poisoned people in their homes maybe 100, 200 years ago. It was also used in painting and fabrics, and it was said to, the arsine gas was released from fungi growing in the wallpaper. And then measurements have also shown biovolatilization from biogenic sources in the environment, either by measuring the volatile emissions themselves, for example, from plants or microbes, or in wet and dry deposition, as I showed you in Esther's work. However, as I hinted at, recent atmospheric models may be missing these important biogenic emissions. So in these last couple of atmospheric models that were performed, they only included anthropogenic and volcanic emissions. So now I'm splitting the graphs in two, while the variability in anthropogenic estimates over time may be due to actual temporal changes. For example, maybe we are getting better for now at decreasing emissions. The spread in natural emissions likely reflects uncertainties in their estimates or potential legacy impacts. So why do we care? Well, as I mentioned, anthropogenic emissions are also changing, at least for Europe. It's decreasing in the sulfur dioxide emission, which also you can see arsenic parallels this trend. And as anthropogenic emissions decrease, the importance of biogenic emissions may increase in the future. Alternatively, if we think about anthropogenic emissions as the entry point into Earth's surface, arsenic can then continue to be recycled, especially via biological processes. But we have to think about what's the impact of the biogenic emissions after it's from an originally anthropogenic source. So how we investigated part of this question or started to investigate was using the next generation model IconArt to simulate global atmospheric arsenic cycling from emissions, chemical transformations, and transport to deposition. The icon part is the global weather and climate model with a flexible framework, so it allows for regional grid refinements, especially because the grid is not your traditional grid, but is triangular grid. So you can really zoom in on different areas of the globe and use the same model for this. And then the ART extension, or aerosols and reactor trace gases, accounts for atmospheric chemistry and aerosol dynamics. So as I mentioned in our other models, there were the anthropogenic emissions and volcanic emissions, and no speciation or atmospheric reactions or chemistry. So the first step in kind of improving this was to include these emissions, these additional emissions in, as well as speciation. And so here are the species that we included into this model. And then accounting for atmospheric chemistry. The one downside to atmospheric chemistry is that, of course, arsenic, as I mentioned, is very toxic. So there's not a lot of experiments for reaction rates or kinetics on arsenic compared to other elements like sulfur or selenium. So actually, one of the few studies that were done kind of only looked at arsenic and arsenic gas decay. So this is what we were able to include in the model, just modeling as a first order decay with a rate constant instead of the reaction rates themselves. And then, as I mentioned, they are oxidized in the atmosphere. So we also included this oxidation step as a test. Here you can see this is just a scenario of doing a point source example to test the arsenic chemistry. We have our initial emission of arsenic gas coming from our volcano. And then to test our first order decay, we had our fraction of arsenic over the total arsenic. So kind of oxidizing in the atmosphere, as you see over time. So once we have that in the model, we then check that off for now. And then we could also go to our next step, which is condensation to aerosols before eventual deposition. So for this, we took our arsenic gas, and then this is a simulation that has another volcanic eruption, so that we could check the values. Then we had our first order decay to arsenic gas, and then we had our condensation onto aerosols over the globe. So then we were able to test condensation in our model. So condensation is checked. Even with the uncertainties in atmospheric chemistry, though, previous models of another trace element, selenium, found that most of the uncertainty in deposition was mostly attributed to emissions. So we have more confidence in emissions. And you'll have to stay tuned for the rest of the results, because integrating this modeling into a next generation framework that was being actively developed took a lot of time. So hopefully we're still working on what the final outcome is like. But we're going to also include, I'll say this for Steve, marine emissions. This begs a lot of questions on what type of marine emissions we should include, whether they come from actual volatilization or just from sea salt or actual other sources. So if anyone wants to chat about that after, I'd be happy to chat. Lenny's group, the group I was in at ETH Zurich, also has students measuring the marine emission side of arsenic emissions from biological sources such as plankton and algae. So you can stay tuned for that work as well. But once the model is finalized, then we can zoom in on specific regions with the same model, especially very dynamic hotspots. I know some of you work on rice paddy fields, and we also know that arsenic is an important threat and trace element in these fields. And it's very interesting to think about, of course, a lot of work that other people also do here on high arsenic in groundwater in the same regions as rice paddies, which is something we know well, where we have rice paddies in irrigated areas, and they coincide with both populations living in highly contaminated areas as well as groundwater. If I showed a map of groundwater arsenic itself, it would look very similar. But another thought that I just want to introduce as we come to the end of the talk is thinking about anthropogenic emissions and also natural emissions after we finalize the mass balance into these source areas, because this also offers another redistribution of arsenic or threat that affects the similar population in these environments. So you'll have to stay tuned for the outlook on this. With that, I want to thank my funding sources. During my ETH work, I was on an ETH fellowship, as well as to those in the group that I was in at ETH. Of course, Britta and our collaborators in Vietnam, and of course, people here at Lamont and also in Australia who really started me on working on all this arsenic and continue to answer these questions in the future with my research group at UW-Madison. So stay tuned or look out for these names in the future. We're working on a variety of different elements, some new elements, some old elements, and in general, trace element biogeocycling in the environment. So with that, I would like to thank everyone who came in person to my talk or is joining in online, and I'd love to take any questions from everyone. Thank you.
You want to ask your question? It's not very soluble, I look.
Yeah. Yeah. Oh, thank you so much, Anna. Oh, thank you. Yeah. Yeah.
Don't know Athena created it.
Hi, Athena.
Yeah.
Can you hear us online? Athena?
No, I don't think they can.
Yeah. Yeah.
The Zoom is probably on the laptop. And
Yeah.
it's not connected.
Yeah.
The Zoom is separate.
Yeah.
No, it should be, right?
It should be.
Yeah. Yeah. Yeah.
Yeah.
Yeah. Yeah. Yeah. Yeah. Yeah.
Oh, yes.
I can also repeat the question. Okay.
Can hear now.
Yeah. Okay. I will repeat the question from my memory, so you can correct me if I miss anything. One of the questions was that these species are typically thought as really stable in the body, especially methylated and also thiolated species. So how are they like in the environment? And also, what is the stability? Is that correct? Yeah. In the environment. Yeah. And how relevant? Yeah. Yeah. And and how relevant are they for human exposure? So I'll start with the relevancy to human exposure. I think I talked about this before with the thiolated species, but now we introduce methylated species before because at least for the thiolated species, they're clearly more unstable to preserve because you can see the transformation. We did like a time series test where we sampled the water immediately as it came out of the ground. So a lot of this groundwater is very anoxic and reducing. So we sampled it immediately and then waited over time. And then we saw that the species started to decrease, the thiolated species, at least the fraction of species that we found. So for the thiolated species, I think they're more unstable in groundwater to the atmosphere than they are probably in the human body, though. Yeah, yeah, yeah. Okay, so Anna says the thiolated species are also unstable. The methylated species I do think are more stable. In particular, that's probably why we found a larger percentage of them in general in all the groundwater. So in all the groundwater we sampled in the end in that study, in the first iteration in 2023, like 64 out of 64 samples all had methylated species. So the question is, could there have been more if we were optimizing for methylated species preservation? Probably. And also in the atmospheric work that was done at ETH by Esther, in her work, she also found that methylated species were very stable once they were emitted. So I guess it's just, maybe it's always been stable, but under considered. That's why I think we should incorporate them more into thinking about kind of instead of the simple or not so simple, but the model of absorption onto iron minerals being only oxy.
So it's a lot of effort for getting the last 10%.
Is there another question? Oh, okay. Does that
Hi,
answer?
I have a question. Oh, sorry, go ahead.
Oh, go ahead.
Sorry, yes. Hi, excellent talk. I'm Adelina. I'm one of Ben's PhD students. I had a question about any ideas why, I think whenever you were showing us the random forest model, the tree thing, so you mentioned that chloride and phosphate were important factors whenever thioarsenates were being formed. Did I get that right?
Yeah.
Yeah, so I was curious if you had any ideas why that might be. I'm genuinely very interested in that. Yeah.
Yeah, thank you for your question, Adelina. Yeah, so first, I didn't interpret these too heavily yet because I wanted to see if the results would first be verified by our sampling, which it was. For the chloride, I think maybe it has to do with the recharge water into each of these aquifers, because also well depth actually wasn't an important parameter here, but we did see a statistically significant difference between thiolated species which were measured versus which were not measured, which I showed you with the violin plots. So those were in shallower wells. So maybe there's a recharge source difference between which aquifers have the capacity to have thiolated species. I need to look more into the hydrogeology of that. And phosphorus well, phosphorus is always interesting because it competes with arsenic for sorption sites. So that could be a reason why. Well, if there's less arsenic oxyanions, then I would have to think about the competition with phosphate. But in general, I think it's a good indicator of the sorption sites that are available for iron minerals. And I have to see, this just shows you which important parameters are there, but not whether an increase or a decrease in that parameter is what's affecting the random forest model. But that's a good question. I'll look into it further.
Yeah, makes sense. Thank you.
Hi, Athena. So nice. Like Anna, too, it's so nice to have you back here and have one of our super students see the beginning of her career, which we know is going to be, fantastic.
Oh, thank
Doing
you.
great work and a great presentation. That's mostly what I wanted to say, but also that I never knew after working on arsenic for 25 years now, over 25 years, that it used to be in wallpaper and in fabric.
Yeah, especially Greenwood. Yeah.
Very interesting.
Thank you so much.
Thanks so much for being here.
The wallpaper at Mount Vernon, if you ever go there, is all made out of copper arsenate. And it emits a lot of arsenic gas.
Is it walled off now?
No. Other questions? Is there another one online? Steve, you have any burning questions about mass balance? Okay.
What do you want to?
Yep.
Okay. Yeah. Anthropogenically sourced arsenic. Yeah, so a lot of it is similar to coal combustion. So that's like anthropogenic when they make the estimates of anthropogenic sources, because coal has a lot of sulfide minerals, which arsenic is typically just associated with. That's where the largest proportion of arsenic, Anna has a comment, comes from.
Yeah, I think that I was thinking in the model that you
Yeah.
showed in the globe.
Yeah.
I was looking at it and I was surprised that in the US,
Yeah.
the eastern part look like higher levels. It's a global model and it's hard to see the nuances for the US. But if you can see it looks greener,
Yeah.
while the West looks bluer.
Yeah.
And so I was thinking, that's weird.
Yeah.
Because normally you would think, there are all the rocks and
Yeah.
dust,
Yeah.
which should contribute
Yeah.
to atmospheric deposition. So I was thinking the Western should, but
Yeah.
then when you said it was modeled as PM 2.5,
Yeah.
then it made sense.
Yeah, this is basically a simulation of PM 2.5. So
Yeah.
I think it's not so precise to.
So that's what I was thinking.
Sources, yeah.
Then it made sense
Yeah.
that the model was probably not really
Yeah.
correct. But that question about the dust, and would that be relevant for this atmospheric model?
Yeah, so
And winds and because for instance, the people working this New Mexico, I don't know if anybody's from the New Mexico Superfund Research Program listening online, but they've shown how important that inhalation pathway is related to dust
yeah,
emissions there in the western part of the
Yeah.
country. So I don't know if that would need to be incorporated.
Yeah.
Because I didn't see it in your model.
oh yeah, I didn't mention it specifically, but yeah, dust is an incorporation. It's also actually already in the global, like this icon art model, dust is already incorporated as an aerosol source. So what we will do is just attach the arsenic also onto the dust as it gets mobilized and transported throughout the atmosphere. Yeah. I guess the hardest part is that there's just not a lot of data on the original concentrations that are in each of these sources, but this will be accounted for in the certainty when we kind of compare it to the deposition measurements.
Hi, Athena. Can you hear me?
Yeah, we can hear you.
Great talk. So I assume that this global model is actually PM 2.5 from coal combustion is what it's mainly reflecting.
Yeah,
Because
this map.
yeah, this map, because that's where all the coal is being burnt. And that's why the
Exactly.
East Coast looks greener than the West Coast, because there's a lot more
Yeah.
coal still being burnt on the East Coast, but it's a lot less than it used to be.
Yeah.
But on the so can you talk a little bit more about the field methods and how you actually tried to preserve these samples to get them back to the field? You filtered them and then flash froze them and then brought them back, or what did you do?
Yes, you mean the groundwater samples, Steve?
The groundwater samples.
Oh, yeah. I can go back to this slide. Yeah, so this method was developed by Britta's group. So she's been measuring thioarsenic species for a really long time. I think you can check out her most recent publications for the method. But to put it briefly, basically, we made this, you can do different iron chelating agents before you flash freeze it. So we use HBD. And then essentially we filtered the groundwater, mixed it up with this iron chelator so that it would take all the iron out of the solution, and then flash froze it in the box of dry ice that you hopefully can see there. And then we sent them to Brita for analysis. So they had to be shipped frozen as well, which is also very expensive to preserve the species. And then they're thawed in the glove anaerobic chamber before measurement on her ICPMS. But I think a lot of her recent students have great methods section on this if you have further questions.
And then the volcanic emissions are what
Oh, yeah.
species are they supposed to be in? Are they
So that work actually was also by Britta. She found that arsine was the primary species that came out of the volcanoes
that
in the
makes
gaseous form. Yeah, not in the particulate form, in the gaseous form. And
That
then
sense? Okay.
She did some, I think that paper did some trapping to trap the volatile arsenic gas. Maybe there's some particulate measurements in there as well. And then she did find a little bit of trimethylated arsine, but she said that it could be related to microbial species in the volcanic environment contributing to a small fraction of that. And I can send you the paper if you want.
Okay, cool. I remember when you first called me up and said, "Oh, I'm beginning to look at arsenic in the atmosphere." So that
Yeah.
it's exciting to see how far you've
Yeah.
come.
Thanks, Steve.
Take care.
Anyone else? All right. Well, let's thank Athena then for taking arsenic global here. We'll call it. And
Yeah.
we look forward to what's next.
Thanks, Ben. Thank you, everyone. Okay.